Okay, so in the last video, we talked about DNA topology, which is one tool that a cell has at its disposal to compact DNA structure down. And the main idea here was that the natural twist of B-form DNA gives it these topological properties, which we can define as the linking number, which describes the twist of the helix itself and the number of times that helix wraps around itself, called the writhe. And because this is a topological property, it can't be changed by deformation alone. And so if we try to change the twist, we're going to get supercoiling in the structure. So we also learned that there are topoisomerases that are enzymes that change the linking number of DNA by cutting, rearranging, and resealing the structure. And these topoisomerases can then be used to underwind DNA to promote formation of supercoiled structures for helping pack the genome down. And they also play important roles in dealing with the disruptive structures that form during replication and transcription, where you have some local unwinding that's going to create supercoils upstream or downstream of the structure that can disrupt enzyme activity. And we ended that video by talking about how supercoiling, that can help scrunch down, say, a prokaryotic genome, but eukaryotic genomes are even larger, and so we need additional mechanisms to fit the genome inside the cell. And in this video, we're going to talk about how eukaryotic genomes are packaged using proteins to form chromatin and ultimately chromosome structures. And so the problem for our massive genomes is like all this DNA could be like a huge, big ball of yarn or something like that. That's a big mess. And what we want to be able to do is not have it all tangled up everywhere, but instead have it neatly organized into a structure that's nice and compact and ordered. And in order to do this, eukaryotic cells employ a rather elegant strategy of wrapping the DNA around what are called histone proteins to form structures called nucleosomes. And there are 5 types of histone proteins that are involved in the formation of a nucleosome: H1, H2A, H2B, H3, and H4. You don't need to know the names of them, just to know that there's multiple versions of them. And the genomic DNA wraps around these histone proteins to form a complex called a nucleosome. So we have this DNA, and it's going to get wrapped around these kind of disc-like structures like you see here to form nucleosomes. And these nucleosomes are going to kind of, as you can see, compact, even just on their own, the structure of the DNA a little bit by wrapping them up. Now, these wrap structures, if you were to unwind them, make the DNA look sort of like beads on a string, and in which each of the beads contains one wrap of DNA around one of these nucleosomes. And if you want to get a nice picture of how this wrapping looks, there's beautiful crystal structures of these complexes in which you can see the DNA going around. And you can see that if this were to be fully extended, the DNA structure would take up a lot more room. So even single nucleosome particles compact DNA significantly. But the other thing about these nucleosomes is that the combination of protein and DNA together enables for these beads on a string to be packaged further into structures that are called the 30 nanometer fiber. So if we were to take nucleosomes, we can actually sort of stack them nicely together in a larger sort of fibrous structure like you see here. And that fiber involves a combination of supercoiling like we talked about in the previous lecture, as well as DNA-protein interactions and protein-protein interactions that help the structure adopt this configuration. And so already we've gone from these fully extended structures to wrapped around nucleosome structures that are becoming more compact. And then once we take those nucleosomes and we start to stack them on top of one another, now we're really starting to get this structure much smaller in size than it previously was. And so the way you should think about the structure of the linear chromosomes in a eukaryote is, Watson-Crick base pairing is going to drive the formation of double-stranded DNA helix, gets wrapped around histones to form nucleosomes, which get organized into these fibers, which then get packaged into the chromosomes themselves. And so that's the picture that you should have in your mind for the structure of eukaryotic genomes. But this raises some interesting questions for us, because the tightly packed structure for DNA is great for storage. We just fit 3 meters of genetic material into a space of 100 microns. But if we've packed it down so much, how do we actually access the information inside? An analogy here is that we need something kind of like the way they store books at the library in the main stacks. I don't even know if people go to libraries anymore, but if you were to go, you'd often find these things where they're called high capacity storage, where you've got all your shelves next to one another, and you can't access all of the books all at the same time, but you have to have some mechanism for which you can access different portions of the collection. And so by analogy, a good high capacity storage system must provide access to the things that are stored. And so how does that apply to the eukaryotic genomes that we've been talking about? Well, the cell has several tools at its disposal to sort of impact the degree of chromatin compaction that you see in different regions of the genome. And so one of the tools that cells have are what are called histone acetylases. And these are enzymes that are actually going to modify those histone proteins that are involved in the formation of chromatin structures. And these acetylation marks have the effect that they actually make the chromatin structures that can form less compact. So if a histone acetylase comes along, it acetylates the nucleosomes. These nucleosomes are less prone to being packed together, and that helps make this portion of the chromatin more accessible and more easily accessed by other enzymes in the transcriptional machinery. And at the same time, the cell can overcompact other regions of the genome that it's not interested in transcribing using histone methylation. And so if a histone methyltransferase, for example, puts methyl groups on certain histone marks, then these methylation marks can be recognized by what are called heterochromatin proteins, which bind across methylated histones to further compact the chromatin. So if you have histones here that each have these methyl marks, then these heterochromatin proteins will sort of recognize those marks and further bundle and scrunch all of that chromatin further down. And so these combinations of regions that are over scrunched down and regions that are more accessible have historically led to the description of chromatin with two terms. So one term is euchromatin, which describes portions of the genome and chromosome structure that are very accessible. So these are places where transcription of genes can occur, and these are marked by histone acetylation, like I mentioned before. And then we have other regions called heterochromatin, which are inaccessible. And so typically we think of genes in these locations as being not able to be transcribed. And these are marked by histone methylation and the binding of those heterochromatin proteins. And what's interesting is this idea of euchromatin and heterochromatin was actually observed by simply staining the chromosomes. In the old days, they would stain chromosomes with these dyes, and they would see that there were bands of dark and light regions in the chromosomes. And this banding, like whether it was dark or light, had to do with whether the genes in those regions were able to be transcribed or not. And so these dark bands correspond to heterochromatin, and the light bands correspond to euchromatin. And I'll add that there are still many unanswered questions in biology as to what determines whether a region is euchromatic or heterochromatic. So there's a lot of really exciting research being done in this space. So if you're curious about it, there's lots of labs on campus, I'm sure, that explore these questions. So to sum up this part of this, the lecture on DNA packaging in eukaryotes, you need to know that the packaging of eukaryotic DNA is going to have a huge impact on its gene expression. You need to know that nucleosomes are the units in which DNA is wrapped around histone proteins, and that this packaging of DNA into chromosome is going to make it inaccessible on its own. And so we have to have ways of unpackaging the chromatin structure to some degree to be able to get access to the genes that are inside. And we found that histone acetylation makes the DNA more accessible, whereas histone methylation makes the DNA less accessible. And so, yeah, as long as you kind of know the overall organization of eukaryotic chromosomes from DNA to nucleosomes to chromatin, and know that chromatin needs to be made more or less accessible in order for certain processes to occur, you're going to be in great shape for this portion of the class. And to sum up, since this is the last part of the lecture, I just want to throw up again that the lecture objectives that you should have taken away from lecture 2. So you should have learned about how DNA is molecularly stable and the advantages of DNA as a genetic molecule compared to, say, RNA. And you'll have learned about the topological mechanisms for packaging DNA and compacting it down, as well as the mechanisms that eukaryotes use using histones and nucleosomes to package DNA into chromatin. So again, if you understand everything that's on these slides, you should be in good shape for the class. And so I will see you next time in lecture 3. Take care.